Hello, my name is Hamed Ektiari from Tulsa, Oklahoma, Laureate Institute for Brain Research. I'm really happy that we have this symposium at OHBM 2020. I was really hopeful that we will have it at kind of in a physical presence in Montreal, but it seems that we are going to do that online. So we have a specific symposium on symbiosis of fMRI and transcranial electrical stimulation. And me and my kind of colleagues that are going to present you some ideas about the interesting potentials with the combination of these two areas are hopeful to be able to kind of motivate you to join us in these really interesting areas of research in basically human brain mapping. So just to give you an introduction, and we talk about transcranial electrical stimulation, we are talking about delivering a really little amount of current for something like kind of few milliamps in the direct current or alternating current. And so we can have transcranial direct current stimulation, transcranial alternating current stimulation, or sometimes random noise stimulation. When we talk about symbiosis of fMRI and transcranial electrical stimulation, there are basically some major challenges or major potentials in the field, I should say. At least I'm going to target some of the kind of major challenges or major potentials, three major items I'm going to cover today. So hopefully you will have an idea about what are the interesting potentials and challenges in the field. When we talk about using fMRI in the kind of the tES or tDCS trial, we are talking for most of the studies, we have a mechanistic kind of question in terms of, okay, if you do fMRI before and after, how can we see what is happening inside the brain by using brain stimulation? So that is a mechanistic question that we have. But there are studies that people might also kind of try to use fMRI as a predictor or as a response that is going to be predicted. So there are some prediction studies that people try to see if they can predict what is going to happen with basically with tDCS or tES using fMRI. And also we have the third group of studies that people try to inform the electrode montages and the way that they are going to stimulate based on the fMRI data. So we have another group of studies trying to use fMRI and that way. So we basically have three major types of integration of tES and fMRI. We have recently done a systematic review with a brilliant PhD student, Peyman. Peyman has done a really nice job on kind of evaluating all the published studies that we have in the field. So we had collected 138 original papers that composed of 220 different experiments of combining basically tES and fMRI. As you can see here, we have 202 studies that are basically mechanistic in sense of using fMRI to see how tES or tDCS is changing the activation inside the brain. 163 with direct current and 39 with alternating current stimulation, okay? Including the studies that we have done in terms of showing that, okay, how tDCS could modulate brain activations using fMRI before and after. So I'm not going to go to the details about that. But these are basically the mechanistic studies. But when you talk about these mechanistic studies, you really like to know, okay, what is the conclusion of these mechanistic studies? When you target a specific area in the brain, how basically tDCS is changing the activation inside the brain. Two postdocs from Michael Nichel lab, we have done a collaboration to see, okay, if we go to all those studies that I told you before in the systematic review, what are the kind of the changes that we have inside the brain? As you can see here, when we target a specific area in the brain, there are people reported that there are activations or changes in the fMRI activations or connectivities within different areas in the brain. Of course, I mean, there are reports that when we kind of stimulate a specific area in the, let's say, prefrontal cortex, we have changes in the prefrontal cortex activations or fMRI signal. But it's interesting that especially for the prefrontal cortex, we can see that there could be kind of changes in the activations in other areas in the brain. And that would make things really interesting in terms of, okay, when we talk about brain stimulation, especially when we talk about prefrontal brain stimulation, we are also talking about modulating subcortical areas based on the connectivities that we have with these areas. Those who are interested to have more details about the systematic review and kind of the results, we have recently put that in medRxiv so people can go inside and see the details about the results that we have. But based on the number of the studies that I told you, you might say that, okay, it might be good to do a meta-analysis to have a better sense about, okay, how direct current stimulation on a specific brain area would change the activation inside the brain. So we had 163 mechanistic studies with tES, and 71 of them are on basically sensory motor area. So they put the electrode over the sensory motor area. So that is a kind of reasonable number to be able to do a meta-analysis using different meta-analysis way that we have with neuroimaging and fMRI data. But when you go to the details of these studies, you can see that there is a huge variation in terms of the methods that people have tried to use. People have used task-based, resting state, different intensities of current stimulation, different variations of duration, different methods of applying the current. So it's really hard to find two single studies that they replicated the results, because I mean they have used really different variations. So it's really hard to do any really high quality meta-analysis or any sort of conclusion in the field how tDCS is changing the activation in the brain, even on the kind of montages or kind of targets that we know that we have many studies inside. So we realized that we need to have a level of both harmonization, also transparency, because it seems that people are not sometimes reporting all the details that we need to be able to conclude. So we moved forward and we made an international network of tES fMRI, basically labs that are doing tES fMRI. We sent an invitation to a group of colleagues, and we ended up having over 50 labs across the board joining to the network. And we are running webinars, kind of different sorts of meetings together. So it seems that we are pretty much in a good shape in terms of establishing collaboration on that level. So if you are interested, please just send us an email or find us on Twitter, and we definitely would be happy to kind of help you if you want to set up a study in the field in a kind of a scientific rigorous way. The second issue that we have in the field is we are facing with a kind of huge level of inter-individual variation in response to kind of electrical stimulation inside the brain. We know that when we talk about kind of head modelings and the level of current that we have inside the brain, as you can see here, we can have a variation in terms of how much current we can have in different parts of the brain. So we have different dosage of current in different individuals. This is one of the studies that we have done here with 65 subjects. As you can see here, we have a huge variation in electrical current distribution inside the brain. So the question is, okay, how we can inform fMRI based on the electrical kind of dosage in different parts of the brain, and how we can integrate these two sort of kind of brain mapping modalities together. So we can have integration in different levels. We can have integration in the whole brain level. So we can, within each voxel, for each voxel, we can integrate the data. So there are challenges for that, but that is one thing that we can do. We can also do what we call region-based integration within specific ROIs. We can think about how to have a specific integration within the clusters of activation in the fMRI data. Or we can even think about network level integration of the data that we have from the computational head model in terms of the dosage of electricity that we have in different parts of the brain in combination with what we have in the larger scale network or smaller scale networks in the fMRI. Just to give you an idea about one of the kind of potential things that we can do, just two examples, as you can see here, with two different montages, F4, F3, F4, FP1. So we explored how we might be able to integrate the computational head modeling in the subject level with the fMRI data, with also kind of the network-based level data. Another really brilliant PhD student, Ghazale, has done a really nice job putting these things together in both larger scale networks and also in the nodes of different networks, and kind of explored how we can do the integration and what are the networks that would be affected more or less with the stimulation, different stimulation protocols. Those who are interested, Ghazale is going to present her results in one of the kind of poster session, poster number 17, so you can go and see the details that she is going to provide. If you want to have more details about some other case studies that we have done on integration fMRI and computational head models, you can read another paper by Zainab, one of the PhD students from Maron Bixon team. She has done a really nice job in putting together details about that. So you can read all those details on that publication, Human Brain Mapping. The third issue is also really important, is about how we can optimize the stimulation protocols for each individual subject. And that's another challenge that we have in the field. So we are running right now a trial, what we call a frontoparietal synchronization. So we try to increase the connectivity within the executive control network by using alternating current stimulation, synchronized alternating current stimulation between nodes inside that specific network in the frontal and parietal areas. But one of the challenges that we have is about what would be the optimum frequency, or even sometimes what would be the optimum phase difference between these two basically electrical stimulation that we are applying in these two areas. As you can imagine, there would be a kind of large parameter space when we talk about different phase differences, different kind of frequencies, and that would make things really complex in terms of how to find the optimum kind of stimulation protocol for individual subject to be able to make the highest level of connectivity between these areas. We recently developed a new setting on being able to do concurrent electrical current stimulation inside the scanner. And we have done different testing recently. It seems that I was really skeptical in the first days, to be honest, but it seems the result is much better than what we have been expecting before. So right now we are moving forward to do what we call a closed loop tES fMRI. So what we do, we basically measure the connectivity between frontoparietal nodes, and then we stimulate, and then we measure the connectivity again to see how we can find the optimum stimulation protocol to do the stimulation. And in this figure that is provided by Lorenzo, I mean, people in the Imperial College, they have done a really nice job in that field. Some really preliminary studies, but it seems that field is expanding. They are using kind of large pads of electrodes. We have recently developed really small electrodes to be able to do the same, let's say, sort of focus stimulation inside the brain. So this is another development that we are working on that. Another brilliant PhD students, Benny from Oklahoma University, is kind of doing his thesis project on that. We have collected one subject right before the pandemic, so the center is shut down right now, but we are just getting ready probably in the next 2-3 weeks to be able to be back and run this experiment of closed loop resting state and transcranial electrical stimulation together. But as I told you, there are some challenges in terms of doing concurrent tES fMRI. There are also really great potentials for that. So we made a specific collaborative network of labs that are interested to work on concurrent tES fMRI. And we developed a methodological checklist in terms of the details that you need to pay attention to when you want to run a trial and that quality. And those who are interested, they can find the details about the checklist on OSF. You can just search for that. There are some details inside, and those who are interested to collaborate, they can just send us an email, and we will definitely be happy to help for that. So just to conclude, it seems that there are really interesting, exciting potentials for combining transcranial electrical stimulation and an fMRI, and there are definitely lots of challenges that we need to kind of handle in the future. I think that we are still in the kind of first step to have a better idea about how polarity, intensity, and duration and montage of electrical stimulation could basically change what we have inside the brain. I'm going to refer you to a paper that we published in Neuron about those challenges. And I think that the paper is still really helpful for those who are interested to start to have a better idea about potentials in the field. Okay, so I'm out of the time, so I need to kind of thank my colleagues and collaborators, especially Michael Nietzsche, Maron Bixon, Vincent Walsh, Filippo Fregni, Martin Paulus, those who basically help us to be able to run these studies, and all those brilliant PhD and postdoc students. Thank you very much.